the high demand from various segments of the country, like from various universities, there were a lot of registrants. We, at the last month, we circulated a Zoom link for those who cannot come in person to these places. But I hope everyone could get the benefit. Those who are here could meet him in person and get your queries clarified, have a chat with him, and get productive input. Now, if we uh, look at the profile of the resource person, Dr. Chathura Gunasekara is the resource person today. Bioinformatics programmer and an analyst from the Baylor College of Medicine, USA. And he is the BA Computational Science graduate of the University of Colombo and has done his PhD at the Michigan Technological University, USA. Current research interest is to unravel complexities, epigenetic regulation across diverse human populations, understanding how DNA methylation variation contribute to differences in disease susceptibility and health outcomes. And information how as you might know, we have global digital platform of overseas Sri Lankan scientists and professionals, which is a database, and the Chathura Gunasekara willingness of contributing to our motherland got registered there, and during a brief visit to Sri Lanka, he was willing to conduct a session at our request at the NSA. So with that background, I would like to invite Dr. Chathura Gunasekara to start the session. Over to you, Dr. Gunasekara.
Thank you for the introduction. So first of all, I want to thank you for giving me the opportunity to contribute to science in Sri Lanka. As mentioned earlier, I studied University of Colombo. I did my bachelor's degree in computation and physics, and I went to USA to do my PhD. And if you look at my background, there's no biology in this. You might be wondering how might be in bioinformatics, right? So I will tell the story first and get you up to how I came to this position. My bachelor's degree was like trying to find a position in computational science. And at the time, bioinformatics like very early. Like that was it was about like 11 years ago, 2012, 2013, and I got involved with this big research lab in Michigan and did my PhD. My thesis was in developing bioinformatics tools and algorithms for identifying pathway regulators in gene regulatory networks and visualizing gene expression data. I was able to publish several papers during my PhD. So this is my first introduction to bioinformatics, and I spent about 4 years studying this. And at the time, there was no special programs to study bioinformatics, so people from different departments collaborate with each other to do this research. After my PhD, I did postdoctoral research at the Baylor College of Medicine, Houston, Texas, and there I was able to contribute to the field of epigenetics, which is like a very new field in genetics. It's like the cutting edge of genome research. I will have more details on this later on. For going that, I won't give you background of bioinformatics. And I want to get some idea of in the audience. Have you heard bioinformatics before? Have any experience with it? Just give me a show of hands. If you have any, at least heard of the term. Okay, so there's some in the audience. are there any expert bioinformaticians in the audience? No. Okay, good. So I tried just my slides to very basic, like very beginner focused way. So I hope it will help you to at least get up to speed with this field and propagate this to others, colleagues, and start this field in Sri Lanka. So at the time when I started my PhD, bioinformatics is like this intersection of mathematics, statistics, computer science, and biology. So people from computer science department, like I was, collaborate with somebody in the biology department. And when we have statistical problem, we can consult a statistician in the statistics department. And we collaborate this with this collaboration, do the research in bioinformatics. That was like years ago. And after that, like last couple of years, there are so many programs that specifically developed for bioinformatics in US and other countries. Yes. Okay. Stop.
Oh yeah, now we can see.
Screen or the screen or the screen. Press W or the
Yeah, now we can see.
So at the time, so the bioinformatics is like different collaborations between different departments and people from biology department when they have computation problems, they collaborate with computer science people and then collaborate with statisticians or mathematics. So the bioinformatics field was developed early on. And I had about 10 years ago, this is the overall overview of what bioinformatics is. And fast forward to today, now it has like matured far, now it has like developed into like its own field. Now it's more like a data science field. Bioinformatics is data science that investigate how information is stored within and processed by living organisms. So that's like the definition of this bioinformatics field, and it's like now entire field. And if you look at these three circles, you can see they are in bioinformatics, they are computer science problems, and there are computer science experts and mathematics experts, and they are also domain knowledge experts. In domain knowledge, you can see like there are specific areas in biology that need experts. So the people from other departments can collaborate with them. For example, cancer genetics is like completely different people from DNA methylation, who study DNA methylation and epigenetics. So people who study microbiome, that's a complete separate field, and also biology and so forth. So this is like the picture of bioinformatics. So if you are coming from background in data science, you can easily collaborate bioinformatics or biologists and can go into this field. And to explain why we need this field, I want to go into a little bit of history. Like before 2000, the bioinformatics is mostly focused on like studying certain regions of the genome, like studying a certain sequence of the gene in DNA sequence, and very targeted approach. And after 2000, it's went into like this, looking at this entire picture of the genome, and that corresponds with this new technology called next generation sequencing instruments, development of those technologies, and also the computing platforms, the the improvements of computing platforms, because these data are very huge. Like usually, if you sequence the genome from human, it's like about a couple of terabytes. And if you analyze the data, you need another couple of terabytes to analyze this data. So it's like, just look at the storage alone, that's a huge increase. Can't do that before 2000. So this has to happen along with the development of computer technology. And if you look at the cost to sequence the genome, about 2002, 2003, that's about $1 billion. It took about $1 billion to sequence the genome. And it dropped significantly over the last 20 years, and now it's about like $1,000. So $1,000 is still a high price, and because if you see the genome, you're not guaranteed to get everything out of it. So it's a high price. And for example, for a developing country, you want to sequence like 100 or a couple of people, it's going to like $100,000 or more. So it's very expensive still, but it has gone down significantly over the years. And so this gave access to many other parts of the world. Even though $1,000, there are so many resources available generated from the US and the Europe. So other countries can at least get into learning and experimenting with these sequencing and technologies surrounding that. First, I want to give a bit of background on DNA sequencing and what it means, because it's a very broad field. If you look at DNA sequencing, we know our cells have DNA, so DNA sequence, and sequencing, we read out each letter of the sequence. So DNA sequencing is the catch-all terminology or phrase that describe the process of identifying the composition of the DNA molecule. And we know every cell in the body has a DNA sequence. So when we sequence the DNA, we'll know the composition of those DNA sequence. And what's the job of the bioinformatics? Is to analyze this data and get some information out of this. So usually the data comes in this format FASTQ. Those are like just text files, text-based files. So arranged in text files that all they have is nucleotide, like DNA sequence information. And we have to analyze this data and get the information out. So that's where basically most of the bioinformatics starting position after doing the DNA sequencing. There are some involvement before DNA sequencing, like design to experiment to sequence which samples to sequence, like how many samples you need. Those kind of questions that happens before the experiment. Those things, there are certain involvement bioinformatics person to like detect, like calculate how much samples you need to get certain power to find difference between two individuals, two people. Those kind of things, there are involvement in bioinformatics, but mostly happen after getting this data. So this data is like very like complicated, and you need specialized tools to run this. So as get to a point that you like special people to study this software and how to run these things. So what gets sequenced? So sequencing is a complicated process. So there are like, when you take a tissue from somebody's like human body or like a plant tissue or something, and it has to be transformed into format or a process called library preparation that then gets into sequencing. And so that library preparation process is like different from each different technology. For example, if you want to study about epigenetics, you have to have a different library preparation technique to do the sequencing. And if you want to study RNA sequencing, that means how the RNAs, how much genes are expressed. So that's a completely different library preparation technique. And so it has to like so once prepare the library, you lose some information, you might gain some information. It's like there's this in between process called preparation that needs to be in laboratories. And when we sequence, as I said, we sequence the genome. So what's a genome? So genome is, the entire read of DNA sequence in our body. And so this genome has reads that contain certain instructions to code for genes. Like, for example, if you certain person, certain eye color, there's a gene for that in the genome. To read the genome and then that correspond to that certain eye color for a particular person. So that's the genome, and genome has this information in it. And in addition to genes, there are other factors that regulate this gene. And so those, like everything is arranged in this gene. So when you sequence the genome, you get all the information. How big is the genome? For example, the Ebola virus is about 18,000 base pair. When as base pair is like this, one base pair is one nucleotide. It's like 18,000 base pairs. If you look at the E. coli bacteria genome, it's like 4 million base pairs. And if you look at the human genome, it's about billion base pairs. And the salamander species about 120 giga bases, that many base pairs. So there are different sizes of the genome, depending on the size of the genome, have complications because you have more sequencing data to deal with. So I'm giving here a little bit of background so you can follow along with me in the next step. So this just to give what we sequence in the genome. And also, the thing that you need to know is the gene. As I said earlier, gene is a certain region in this gene. And it's the official definition is region that includes all of the sequence elements that necessary to encode the functional transcript. That means it serves a particular function from that sequence readout serves a particular function. When a gene is transcribed, we said the gene is turned on, means that gene is like working and doing something. So the DNA, like blueprint of the gene, and we make a copy of that, and that messenger RNA, it's called the messenger RNA, that's going to do a certain thing called a protein. So protein is another compound that binds to DNA and does some function. So when we do bioinformatics research, we may work in the DNA level. That means we look at genome of a particular person and we compare genomes between people and identify certain differences. The genome, like one people has this sequence location. For example, if you have T in one location and the other person has T in the location, that difference might contribute to something like disease or like different phenotype. So that's a DNA level. We study bioinformatics. We find those differences. Also, we look at the RNA level. Like when we have genes, for example, human, there are about 20,000 genes. And we can look at how these genes are expressed. So expressed means how these genes are like turned on. And we can see some genes turned on high level and some genes turned low level. And you can study the gene expression of certain like a tissue, and that's a different way of studying about this. And that's also analyzing it's called RNA-seq analysis. That's a different part of bioinformatics. Another part is the protein level. That means when from the DNA molecule transcribed to mRNA and it does a certain function and produce a protein. Proteins are basically the building blocks of life. So it proteins bind to certain things and it gives something. So some compound. And that level is another level of studying about how this genetic material passed from the genome to a protein there where it does some kind of work. So bioinformatics is involved with all these processes. And so certain labs study DNA, some people lab study RNA, and some people study protein, and some people study even like beyond protein, like when the like metabolic effects, like how these interact with metabolic of your body. So this is like a basic background of where bioinformatics is and studied. So are there any questions so far? Like, if you have questions, I would like to get early on because I know you are understanding this, or I have to go faster. Are there any questions? Yes. Yeah. Yes. For example, if we're studying like the gene expression, you don't need. Yes, it's a different. So if you study DNA methylation, you have to have a. So the question is like when you have laboratory preparation, do you can study like the same like different properties from the same library? Is that okay? Okay. Okay. So like if example, if the library is prepared to study DNA methylation, then you have to treat the sequence with particular chemicals that only measures the DNA methylation in the instrument. So then you can study DNA methylation. You can study anything else on that. I mean, you can, but it will be complicated. So that's how the question. So I hope if you have any questions, I think you can use the window also. So I would like to get questions along the way. Then I can gauge you are getting this or not. Then I can go and forward. Okay, so today, so since bioinformatics is very broad field, there's no way I can cover everything in one day or in in 2 hours. So I want to like get like little bit of uh, background how and certain analysis is done. So in this, I'm showing the gene expression analysis. For example, if we have let's say you obtain certain tissues from these 8 individuals, and there are 4 individuals you treated with some kind of a drug, and other 4 individuals you didn't treat it. So you want to see how the drug affects your genes, right? So that's basically the finding the difference in gene expression because of the influence of certain drug. So then we do this called gene expression analysis. After we obtain the tissues and we after do the sequencing or some kind of quantification from the DNA, we can get this gene expression profile. So the individual get a gene expression profile because everybody has this 18,000 genes. Let's say for this individual has get certain value here, like value here at the other gene, like for the all 18,000 genes, you get value. And likewise, you can get those data for all 8 individuals here. Here only showing an example, of course, because usually this is like hundreds of samples. So this is for illustration. I'm just showing for here. Okay, so I have a question. So I was talking about the of the genome. I said 3 billion for human, and salamander it is like 170 something, 220. So it says, does that mean salamander just more functionality than human? So is the genome size correspond to the intelligence of the human? No, right? So uh, there's there's no really functionality. It's just the size. There are so many like DNA in the sequence that does not do any function at all. There's so many nucleotides that just they repeat itself. Those things that's added to the genome through the evolution, but it doesn't serve any function. So yeah, the size of the genome does not correspond to any more functionality than other animals. So it's not that really correlated with each other. Yeah, so if you like even in humans, like if you if you're talking since you're talking about the genome, so even if you look at humans, like 99% of genome is across everybody. Like doesn't matter you came from which part of the world, like 99% common. Only there's 1% is different between individuals, even within humans. And so they have to show that also. So it was like 20, yes. Yes. Right. Okay. Yeah. So the question is like where is the data public available? So how can we access it? So I have a separate section dedicated to that entirely because that's I think the value is in this presentation. So get to that later. So there are some websites that particularly from NCPI, National Center for Bioinformatics Information from the US government. They allow publicly for everybody to download this data and analyze. We'll share that later on in this presentation. Yes. So first, the question is like I showed earlier, there are different type of files that bioinformatician works with. So there are FASTQ files and BAM files. So FASTQ files are just the raw data, like just raw sequence. So BAM files, you align the straight sequence to the reference genome. So once you align it, measure that information, you just district other information. So FASTQ files are raw data. So BAM files are like processed intermediate files. So you work on those BAM files in this analysis pipeline. Okay, so RNA sequencing. So this is like usually like the data metrics that we work on. So we have certain individuals with treated with something and certain individuals we didn't. So we see that which genes are different between these individuals. So I have some kind of demonstration or like a yeah, I'll get to that maybe after a couple of more slides. So I said about DNA, and are there other regions in the genome? As I said, like there's some regions called untranslated regions that doesn't do anything. I mean, so far we don't know probably, and we say they are just DNA doesn't necessarily function, but we don't know. And there are some regions in the genome; those are untranslated regions. Also, there are regions called promoter regions that certain proteins get bind to the promoter regions that then initiate the gene to turn it on or not. And there are other regions, so CPG islands. If you look at sequence, there are so many CG locations. So if they like concentrated certain region, those are called CPG islands. So there are certain features in the genome that we study in labs, and like some have entirely dedicated their like full research mission to study about the CPG islands and promoter. So there's so much things to study in the genome. So how bioinformatics is practiced? So from what I said so far, it's mostly like data-driven disciplines. Like it's things done with the data. So when you have data, you need computers to analyze, and you need code for software to analyze these things, because you can't do this in manually. It will take so much time, just waste of time. So you have to write code, and software is one of the main component in bioinformatics. Data management, we need access the data from public repositories, and then may have to combine with different other data sets, and have to do the conversions. Because if you do, some people, if there's another lab did a certain experiment in a different way and another lab did different way, but they are somehow, they are different conditions. When you study them together, you have to figure out how they are like certain biases are not introduced in this other lab. And this example, like say time I was studying breast samples, and when I tried to plot those samples in a 2D diagram, I identify certain samples are completely separate from the others. And I couldn't figure out why this breast cancer sample is different from the other. And we reached back, and it was like 2,000 samples, and there's 500 samples are completely separated. We thought maybe it's a complete like different subset of the cancer, or there's so many things coming up. And when we called back the original creators of the data, they said, "Oh, this was generated in a different institute." So that's why the difference is. So there are so many things that you have to care about when you analyze this data. Uh-huh. Uh-huh. Yes. Mm-hmm. Yes. 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 So yeah, I mean, if you look at the sequence, it's all great. But when I say, like, say, when you make the gene expression after the instrument, like this raw data, right? So you run the instrument in different countries. Like when you run the instrument, you might have different parameters set to that. And if you run the same sample in different country, another country, there may be some different library preparation technique is used. That might introduce certain bias in the end result of the data. So that's what I say, like there's some batch effects of the data that you have to take it when you combine different data sources. So I mean, it's not like if you have the exact same thing done in different countries, it should be exactly same, because these are like run through computers. There's no ambiguity in computers, right? So they have exactly the same thing, but when you run the experiment, when you run through this preparation or any kind, because it's a very complicated process, like so many steps involved, there's certain things can change and change data down the line. Okay, so data management is one crucial part of bioinformatics. And then after doing data management, then the primary data analysis. So you have to convert those sequence data. You can't analyze those data by just looking at them. You have to align them, then call the variance. I mean, say variance that like when we read the genome, we don't look at the entire 3 billion plates. We only look at this certain, only the small part of the genome that are different, right? So you have to like run the software to identify only the parts. So those are like primary data analysis analysis part of bioinformatics pipeline. After analysis, then the data interpretation. But usually it's like you do the publication, you publish, and maybe many reviewers review this, and then everybody agree this will be a publication, and that will be either in the one like specific knowledge, right? So that's the interpretation part. So this is basically like the basic bioinformatics pipeline, and what these are what bioinformatics basically do. If you want to go into more detail, I have had here you acquire data, maybe it's based on like completely new DNA, or maybe just download a big data set. And this is the early part of the bioinformatician's workflow. So get the data, you have to organize them, and the annotation filtering is also a part, because these instruments are not very precise. Like sometimes maybe errors. So if there's errors, we try to remove those. Like basically the bit faults. Like when you say when the sequencing instrument call this nucleotide in COG, it also associates accuracy, like a probability, that one, like how good this prediction is. Like very high, we take it. If it is low, we just discard it. So those, like cleaning the data is so important. And after that, analyzing, mining, modeling, do whatever you need to do, get some information out of the data. After all that, then is the publication part, where you interpret the data and then produce manuscripts, figures, and publish. And it will be in the science domain. Okay, so now what is this? It says what is the recommended computer for bioinformatics? I said, like, this is a very computer intensive field, so you need computers to work on. So, I recommend like a Unix-based computer for work on as a your day-to-day computer. It's good to have Unix, because most of the bioinformatics software is developed by the open source community. So they are like open source software. They are freely available. They are like paid software, and like similar to Linux and the Unix operating systems, like uh, open source, these are also like that. So they are developed by the community, and so academic institutions and such that. So Linux is like the best platform to do that. And so I recommend, if you practice bioinformatics, just the Linux computer, and that should be enough to do the basic things. For example, if you want to learn how to do kind of RNA-seq analysis, would be enough if you have about like the GB RAM or 8 cores of 8 cores. So the next question I get is like, does learning bioinformatics need a massive? The short answer is no. But if you want to do some real work, that if you doing something like real analysis done, you need this high performance computing clusters. So these are like computers with like thousands of GPUs and about terabyte of memory. And so usually it is run by one big institute. Within that institute, certain groups request resources from the server, and you get allocated. So let's say you need to run an analysis with let's say 8 cores. You need 120 gigabytes of RAM, and then you do the request. You get that allocated. You run your analysis on, and that will significantly improve the analysis, because usually data like several gigabytes big, these data files. So you can't load these things to a normal computer, because you don't have 120 gigabyte RAM in a computer, right? Yeah, if you want to do like real bioinformatics work, that's like possible. You can publish, but like if you have like real world data, like hundreds of samples, usually the resources runs out in desktop or a computer. So the currently, but so the option is to have the cloud. So cloud is available. It's like Amazon Web Services. You request need these resources, and it will be a high performance computer. You like request those resources. So that's one option if there's no already infrastructure available. But uh, yes, yeah, yes, okay. Yes. Yeah. So even in like I live in Texas, so Texas has all education institutes get access to this high performance computing cluster. So anybody can get account if in a like university in Texas, you can get access to this cluster. So it's like a public infrastructure. Basically, public infrastructure, you request access resources from them, you do analysis, and then you're done with it. So that's like the best option. Right? Cloud, you can go to cloud, but it get really expensive really fast because if you want to store like a couple of terabytes, it will be very expensive. So I wouldn't recommend this go in this approach because still it's very expensive. Best option is to run an HPC cluster. I mean, these are not that expensive. You can uh, combine certain computers together, like 10 computers together, you can build a cluster, like even in Sri Lanka. It's not that expensive. That is most cost effective way because then you share the resource with many researchers in the country. Yes, so HPC cluster, how it works, so it is like a one shared uh, resource, like electricity. Electricity is shared by everybody, so you use it and then you're done with it. So it's similar to that. HP clusters should be available to do bioinformatics if you want to do real work. So job submission, like you request how much CPU, memory, runtime you required, and you request job submission, then you get allocation to run analysis in there. And so that's about PC clusters. Okay, so do you need to learn Unix to do bioinformatics? Yes, so you should learn some Linux and Unix. So this is now it's easy. We have so many resources, and I even have in the end I have some resources. It's pretty easy. Only thing is you see what you are doing like in the command and you have to see the commands. And it's easy to do command line bioinformatics than in the GUI because there are so many files to have to work with. There may be hundreds of files, and in the analysis there may be 10 query files that generated like 100 files. So it's very difficult to work with so many files in a GUI environment. Like basic stuff like Word, PowerPoint you can do, but when you have so many files to work with, it's easy to work in a command line environment. So I recommend learning Unix. If you get a Linux computer, you should learn how to run the command. And do I need to learn programming? Yes, you need to learn programming, and nowadays it's pretty easy to do programming. To say like these so many resources to learn about the programming languages, and there are so many online communities. If you have for example, if you have error, you just need to search the error, and somebody else in the world already had that error and is there. So it's pretty easy this to learn programming, and I recommend doing R or Python. So anybody in the audience doing R or Python? No. Okay, so yeah, the R is recommended for bioinformatics because most of the bioinformatics or biostatisticians in the world develop package in R and publicly available, free available. And R is like recommended package or language to start. And Python, if for example, if I write my own software, I would do Python. But if I want just do some simple analysis, then I do R. So start with R and go into Python. So you need to learn some kind of programming language to do the informatics. And there are certain tools that you can do on like completely like a website. You can upload your file and then the analysis is done and you get the results downloaded. So even I was involved in developing some of those tools, uh, but those are somewhat limited because you can't upload so much data. It's very difficult to upload them. If it's small files, you can, but if you have like gigabytes of files, you can't upload them. Easy to run them locally. Sometimes there are restrictions uploading data to the public cloud. Like for example, in US, you are not allowed to upload the genome sequence data from like people to cloud because this is considered as private information. So there's you have to run your data in-house with your institute. Okay, installing software. So I will share these slides, and you don't need to like memorize. I'll share these slides, and you can like do them back later. So R or Python, when I was starting out, the nightmare was like managing these different versions of software, because one group developed one version for this particular version of Python, and another group developed a different version. When I try to show them together, everything crashes, and it was a nightmare 10 years ago. But now it has gone significantly, like it's much improved if you use these Anaconda package managers. So these are software package managers specifically designed to do in data science and scientific computing environment. And this ability to set these things in computer and get up to speed. And when I was starting, the hard part was getting started the software. It's like was a nightmare before. And for the development environments, like like if you do programming, you have to use some kind of notepad or something to write the code, right? So there's Visual Studio Code I recommend using, because you use this AI generator. So in AI generator, what you have to do is just tell the AI program what you need to do, what they generates code section, and then you can use it. Yeah. So Visual Studio Code is what I recommend to write new software, or if you want to like arrange your analysis in a certain way, then you can present to somebody. You can have your information like text information and so code, and also figures and plots, everything in one, called Jupyter Notebooks. I will show them after the slide. I want to like do some demonstration to show them these tools. Also, RStudio is the platform here you use R code. So all these tools are free, and you can use them to the informatics part. Sometimes you will get problems when you run the software and installing, so it's mostly solvable by good search. So it's all you need is like dedication and willingness to learn, and to like figure things out. It may take like a day or two. So it's sometimes some bioinformatic problems take like a day to figure out a certain error in your code, and that's how it is. So you need to like have tolerance to solve these bugs in the program. Okay, so I want to demonstrate one example. So this data. So this data is available in Geo. So Geo is like a database, gene expression omnibus. So if you search with this ID, you get the data that was published with this paper. So this paper is RNA-seq transcript profiling identifying CRISPR-Cas2 as a so this is a certain like treated with this drug, and how this drug change the gene expression in these tissues. So this is like a small data set. I just want to show you how it works. So I made it like very simple, so it will run quickly in the time. Let's see if I can. So this is RStudio. So RStudio is you write R code. RStudio and R platform is like also the open source community. So they develop this statistical analytical software, and freely available for anybody in the world. So for example, library. So I installed this previously because it takes some time to install over the internet. So I already installed it. So EC2 is a software package that used to find differential expression between control group and the case group when you have a set of genes, like the same. Remember the set I showed earlier, the data matrix. That kind of data matrix, you just need to use this software, so it will run it. So all these tools are developed by open source communities. So I already downloaded this earlier, the earlier. So I will just run this to. So this will load the libraries, and then I will load the data. So this loaded the data. As you look at the top out of the data, so this is, it looks like the top line. If you want to look at the data, it looks like this. So this, each column represents a sample. So they are like eight samples, 4 of them treated and 4 of them untreated. And this is the genes. So if you go down, there are like 63,000 genes in this. And we want to see which genes are different between this group and this group. So that's a very basic analysis you do. So as I said, you load the data. So I will first load the data. Then I need to get some information about these samples, right? You know which one is which. So with this, you can see like this ID started with this is an untreated sample with dexamethasone, and sample is treated, untreated, treated, pretreated. So there are 4 treated samples, 4 untreated samples. And so you need to do some like cleaning to make it run. So all this information you will learn when you learn programming like in. So here is making sure, I will make it a little bit better. So this line, ensure the row names in the column data match the column names in the count data, because we need to put them in the same order, right? Otherwise, it will not figure out which sample correspond to which treated or untreated category. So you need to do something like ordering. So this line does that, and it says all correct. That means we are good to go to the next step. Are they in the same order? Yes. So the column data, the order is same as the row name of the other table. So you two tables match with each other. Okay, so this is like where part it's reading the data in, and after that this function is like from the ESEEK2 package. Somebody else had the write the code for this. It's available in the online. So we use the package and we give our data, count data, and our column data, and give a design. So we know we want to see the difference in the column of like dexamethasone. You look at this column. So you look, the program looks at this column and figure out which samples are untreated, which samples are not treated, and then accordingly run the program. So if you click this, so it's running. After that, so it ran, it gave, so yeah, finish. Because this is like only 8 samples, it finished running quickly. And so here, what I meant by cleaning data, like here, if the rows, if you look at the counts data, empty rows, right? So those doesn't contain any information, right? So we don't clean those things. So we can clean, like if the count is only greater than 10, we keep them, we keep them, and we do it again. And then we do the factor and the DESEEK. So it fits model and we get that the result. Can get a summary of it and can produce upload. Okay, so produce plot. So here I shows each point represent a gene, and the root ones are the genes that are different between the groups. So these are the genes that are actually changed because of the treatment. So in the next stage analysis, we can like isolate this gene and can like look at why these genes changing tissue in this after this treatment. Then we can look at what are the pathways in the biological pathways that these genes are involved, and downstream analysis. So this is basically like a very basic workflow of bioinformatics. Right. So next, I want to also touch upon this point, scientific reproducibility. Especially in life science, it's very crucial. You should be able to reproduce your research like somebody else can reproduce the results, right? So then you can even like review, like if you want to publish a paper, you submit your result. It is recommended like in high impact journals like Nature or Genome Biology, they ask you to submit how your analysis is done, like specifically the code and the raw data. So if they can rerun it and get the exact same results. So with bioinformatics, it's possible to do this because it's you can give the code and you can give here's the raw data, here's the code, here's the output. Anybody can just go through the code and see if we did it correctly or if there are any issues with it. So in terms of scientific reproducibility, bioinformatics is like important, playing an important part. Okay, so I will give you some case studies of use of bioinformatics. And this is one of the good examples, recent example of monitoring this COVID prevalence in certain like areas. Like in Houston, there was this project done collaboratively with Rice University and Medical College of Medicine. So they look at wastewater from different part of the city and look at how the load in that the wastewater, and then from that can see which if it is on peak or not. So that kind of thing done in center. This is an invasive surveillance tool to estimate the prevalence of viral in communities. So they made a really nice plot that with the viral load, it perfectly corresponds to the PCR testing the possibility, and that shows in the paper. Next, like the precision medicine is like one of the most like cutting edge techniques in bioinformatics. That means in precision medicine, you want to like predict where you're getting a disease or you treat a person, you need to look at a genome profile and do the treatment according to that particular person. So precision medicine is called basically like personalized medicine. So that is like a very hot topic in the United States. So that's be done because of bioinformatics analysis, because each person's genome, you have to analyze each person's genome and a genetic profile for every individual to do this kind of thing. So it's like up and coming the field of bioinformatics. And also cancer genomics, you can look at cancer tissues and normal tissues and identify difference in the nucleotide composition, and then maybe develop a certain drug to target those genes that are maybe excessively expressed or deep underexpressed, change those things. And also, this is surveillance and control. Like for example, in a pandemic situation, there are now so many platforms have developed, like from different countries. You can submit it automatically submit the viral gene sequenced, and it goes into one repository. Then it can track with the new mutations in the viral genome. So this is also one of the case studies. And this is the resource and the database section. So this is the one database I showed you. Bioinformatics, you can't do bioinformatics just like looking at it. Like you can do bioinformatics like in point, because you need to like have hands-on experience with it. So the best way to do is just download the dataset and try to run it. So, so to do that, luckily there are these resources, and GEO is gene expression omnibus public repository. It archives like freely distribute these data sets, microarray. Yeah, so those are like kind of the but still gives some information. And so also next gen sequencing data, you already have like the raw data. If you want to process the whole raw data, you can download. You can just download the process data. So you need like extensive computing background. You can run in your laptop. So sometimes they give the the process data as well, and other forms of data are available. So to familiarize with genomic data, it's best way to like find which area you want to study, and then download the data, RNA-seq dataset or chip-seq microarray data, and then start analyzing on that. So yeah, you will have to do so like in R, there are certain pages that are available that you can directly download. So you just need the code or the ID from the paper you want to reproduce, and then you just plug into the GeoQuery in R page, and it just download the data directly to your software. Then going to the level is pretty easy. You don't need to go to the Geo website and find it. And download. So there are certain things that make it very easy now to do the handling and processing. Yeah, so to conduct statistical analysis, you can use these R programs or even like Python. So there are those tools. And so gene set enrichment analysis is then, as I showed earlier, I showed there are certain genes that are different between groups. Once you get that list of genes, then there are already online tools, plug those set of genes, and you can see what the biological pathway those genes are enriched. So then you can see which genes actually change this biology of this tissue. So those things are like already online available. It's pretty easy to do gene enrichment analysis now. Yeah, so yeah, engaging with community is important in bioinformatics. You can like online forums and discussions, and especially collaborations with different departments is also important when you do bioinformatics. And yeah, so these are some of the links. So this is one. We can, yeah, it's okay. So yeah, this first one is like how you organize your data in bioinformatics like project. Like when you have tons of data, you have code, you have data, you have process data, and there are so many intermediate files. You might need to manage those things. This paper is good to do that. And this one is for introduction to R, and this is also R. And this one, bioinformatic notebook. This is good Linux and your background, so you can like just follow along from the beginning, and you'll be pretty not. So this is also 3 good reasons I can suggest. Yes, so I mean I had some like slides in case I ran out of like things to say. So I don't want to go into this before like I want to like do the Q&A and get some feedback from the audience. So yeah, if that's okay with you, then you can say if you want to go more details, we can go in to more cutting edge bioinformatics. So do you have any questions for me? This Lisa?
So,
Mhm. Mhm. Yes. Okay, so do you mean the reference genome, like which reference to which? Yes. Right. Mhm. Okay. So like in yes, so obviously like there are difference in genetic composition, like if you're from Sri Lanka or for like if you compare, yes. Yeah, so they yes, they are good, but if you try to do that, then there's no way we can standardize this, right? So there's certain things we have to standardize. For example, reference genome, there's one reference genome for the entire human, like it doesn't matter which entities you are from. So there are certain things that you have to like uh, have as a reference. So there's one thing is a reference genome. So regardless you like the sequence data from like African population or white population or Asian population, it's the same. So yes, one for RNA. Yes, so it's called HG38. Human genome 38 is like the most recent gene. There are more recent one. I wouldn't recommend going to the most recent because of the other tools are not like developed to that level. So it's always best to go with the stable version. And so it's 38 is the one stable currently. Yeah, it's all right. No problem. HG30, that's the entire genome, right? So it's entire genome. So let's say you in your RNA-seq experiment, you might not measure the same the entire genome, right? So you might measure the exon or intron. Like the exome sequence is a different part. Introns are different. If you will only look at the transcript, you only get that part. You will not get the other parts. But from RNA-seq perspective, you only look at like how is 30,000 genes between individuals, right? So it's only the intron. That translation, right? Yes. Mm. Yes. Right. Yes, it's there. Because it's the genome. When you get the sequence, if you map to the HE38, it should be yes. Uh, So Illumina sequencing platform, yes, that's that's what mostly used in United States and mostly cost-effective way to do it. It depends on like the setup of if the lab is already like prepared to do that, then I think that's the way to go. If you want to change it, it will be like different. Like you have this learning curve how to do that. Right? So yeah, there's like different technologies coming. Like but Luna is like the most like short read sequencing is the most so far. And yeah, that's also in the United States, so in China, like sometimes even from US, we send samples to China. Sometimes it's cheaper to do. And Illumina sequencing is the most from one. Yeah.
So,
Yes. Yes, that's the old standard, but used most of the world. So, but there are different parts. I'm not sure about that's the gold standard. Like for DNA, like for example, epigenetics, there's like whole genome sequencing. So that's the gold standard. RNA sequencing, I'm not exactly sure. Gold standard. Like for example, you have like microarray sequencing that is maybe like more cost effective than the sequencing. So that's like maybe depending on the preference. I don't really know the gold standard for RNA sequencing. So some things they are like for epigenetics or DNA mutation, that is gold standard, but for RNA, I don't know. But I'm not really RNA sequencing. My main research is in DNA sequencing, so I haven't done that. Yes. Yeah. Metagenomics. So metagenomics. Uh, So that's not the area I am familiar with. So as I said, there are different domains in the bioinformatics. I'm not in the metadynamics. So to be honest, I never analyzed metadynamics. So, but if I want, I can show you a machine learning example I did for DNA methylation. Do we have time? Like, I'm not sure. It's 11. Yes. 11:30. Okay. So this is one example of, yeah. So here in this example, I will start with. Yeah, for this, I need to first give you some background epigenetics. So I will first start with that. Epigenetics is for, like we in DNA sequence, we have like the nuclear like this, right? So, and like DNA methylation, DNA methylation, you can see like this, the methylation means like we have like the C position. Some of the C positions have this CH3 part. So that's called methylated cytosine. And so that called it's like a methylated CpG. So if you look at the genome, there are sometimes you have the methylated, sometimes they are unmethylated. So we know like they're with CTG. Methylated cytosine, maybe like the base, like so it's a different one, right? And what's important about this is like genome sequence, we know it fixed, like the cell, it's fixed, right? To take genome sequence from any part of the body, it's the same. But methylation from one place to another. And also methylation can be influenced with the environment. Like for example, during like prenatal development, if there's a nutrition imbalance, there can be different methylation pattern for a certain individual because of the nutrition imbalance in the when it's in the mother's womb. So that kind of that influence the epigenetics. And here I'm showing like a analysis pipeline that we use in epigenetics. So first we do the sequencing and we do quality checks and we do all these like pre-analysis steps. That's doing the quality step. After that, we do the alignment. And after remove PCR bias, so then it will we don't have any bias in the data. After that, we call the methylation calling. So in that we can say we can calculate what the percentage methylation for a certain CPG site. And so this is relevant public health because you can predict somebody's health later on based on the methylation pattern discovered early. So that's why important. And when my research, I was trying to identify regions in the genome that you measure, like in a like prenatal blood, early not prenatal, like blood sports, we can detect this pattern that DNA methylation pattern exists, and we find able to predict certain diseases based on the blood methylation. So that was my research to identify these epigenetic variation, locations of epigenetic variation in the human genome. So, for example, here the methylation in the regions that I discussed, we can like measure the methylation from any part of the body, and you can measure like infant methylation across the different parts. So that's so this is like kind of long topic, so I didn't go into. I'm not going to detail, but I have identified these regions in a research project, so that can be used to predict the population levels across tissues. Question. There's a question in the chat. I will answer that while I go through this. Do we have different algorithms to do predictions or classification? If we have different deep learning or machine learning algorithms. So it's, okay, it seems I already have answered. So like that's the methylation. In the methylation pattern, we can predict certain diseases. So I will back to my. So this is okay. This platform also I want to like show. This is called the Jupyter Notebook. So here I'm running R code. I can't run this right now because I haven't like installed all this software. It will take some time. So we'll just go through this. So this like similar to the notepad, you can integrate your code and as well as your like explanation or anything in there. So that's part of this the Jupyter Notebooks. This one thing I use. So here I showed actually the with it can we specify individuals based on blood methylation whether those individuals have schizophrenia or not. So schizophrenia is a mental disease based on blood methylation. We classify those individuals. That was the problem. And here I have done some of the pre-analysis. For example, here I showed that first I had to show we measure methylation in blood, what does to do the methylation in brain? That's one of the research questions we have to answer. So to do that, I use this public data. So this is data set available publicly. So it's like free of charge. You can download them and analyze it. So this is GSC 9 for this also from downloaded from the G. So then I analyze data in the context of the regions that I'm interested, and I show that like here the red color shows correlation. That means blood first. This is cerebellum is a part of the brain. Blood versus this is another the 4 parts of the brain. So I showed now the regions that we identified. Relation consistent because blood and brain. So using these variables, so in machine learning, you need the variables, right, to predict some case of control. So basically here, what I did was like a classification model. To do that, I use this variable. Let's see. So yeah, I load the data, and these are basically the variables and the status. So this is I want to like predict whether one or based on the methylation pattern. Once we do all the processing, everything in the end, like when we do the analysis, this is what we be working. Bioinformatics analysis, but most of the work is done on to the data into this format. Get to this format is actually easy. So here I actually running a model called least squares discriminant analysis. So for that also I use a certain library that's already pre-existed before. And here I do some like tuning to identify parameters. We'll go to, so this shows I represent this high dimensional data in the 2-dimension. In 2-dimension, you can see like this one means the people who have schizophrenia, and 2 means who do not have schizophrenia. You can see that they're like the separation from the methylation pattern in this plot. So from using this, I develop an algorithm like calculate a certain risk for disease. So from that, we can calculate the risk and we can plot in distribution and see whether this individual has this high risk for schizophrenia. So that's like one machine learning. If I can. Yeah. So yeah, here I'm showing risk for schizophrenia. It red means the case group, blue is control group. This is in the training data. So in the model, our model predict for the people who we didn't have seen before in the model. So new samples, we predict the risk, and we see the prediction is also good. Like it predicts in the new sample whether certain people have this risk for schizophrenia, and we can identify this risk plot. Yeah, it's something here. This was actually published in molecular psychiatry journal. Yeah, there's one example for machine learning application. Yeah, so do you have any other questions? It's more important to like answer questions than share research, because you can just Google me and find out my research later. Go that way. Yes. You mean like 10 years? But I mean, that's the surveillance part, right? So as I showed earlier, so previously, for let's say the coronavirus, we didn't have a surveillance platform that integrated across the world. Like then you can see like the same at the same time, you can see increase in different part of the world. You can see we are in some kind of a pandemic, right? So right now they have developed like a centralized server. So all the sequencing done around the world. Like if somebody sequencing for virus, some sickness, and it happens in let's say China, and also happen in some in the Europe, then you can like see the whole picture around the world. So I think starting now, like because the pandemic, there are certain things that are coming out, but I'm not familiar like something you can predict like in 10 years. Like not sure. Not sure that no. So like same Not sure. I mean, yes, I mean, there can be predictions, but we don't know, right? It's until it happens. No, sure about that. This one for this, so this is completely done with public data. So for all this publication, um, no, we didn't do laboratory tests particularly for this, but I integrated multiple data sources from different labs, and we were to find kind of a agreement in all of those. So then we can say this is actually some kind of signal in methylation that associated with disease. So, but we haven't done like specific biological like this. No. For the data I'm showing. So yeah, for this I think it's pretty. So this is done in different platforms for 50K array. So for 50K array, it's not sequencing data. Yes, for this form, there's no sequencing data. Sequencing. So they directly give the population methods because this measures about 450,000 like PG sites from the genome. And to make it cost effective, there are certain platforms have been developed for like methylation. They are like 450K Illumina 450K platform. It only measures this 450,000. Measures only 450,000 regions in the genome. So that's like if you use that data, you don't have to do it deal with sequencing because that's only measures that. But sequencing is like when you get the entire sequence, like in a gene. So this data, like, yeah. Yes, 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 yeah. For my project, yes, right, correct. Yes, yes, correct. Yes. For but the very early part, I had to do entire genome. That was I did in 2019. So that actually the paper published in 2019. Like so for that I used like 30 whole genome tissues, the whole genome sequence. Uh, in which slide? Yeah. You mean here? Or in in everything here. So here, this is not RNA. So this is DNA sequence. This is DNA sequence. So each like the read is came from one cell in the tissue. You read that part of the genome and take that cell and take that part of the genome and we put them all together. So this is called the alignment. So for this one, so alignment part, there's like this is actually the file or like I told you from the file, you can see these things. So there's like the ATCG lines, and once you align them to the correct location, so it's like very like computer incentive thing. They're like like there are billions of lines, and you align them to this like 3 billion base pairs. So it's like very computer intensive. It will take like to 2 weeks to like the alignment. The computer running weeks continuously to do the alignment alone. So it's like very compute intensive work. So in my research, so like we start with the whole genome and we like trying to find the regions in the genome. So then we have to look everything, right? So but if you're looking at like gene expression, then you don't have to look at everything. You can only look at the gene, 30,000 genes. It's much cost effective. So that's why like platforms develop. If you want to do sequencing, you can do that. Or if you want to do other platforms, that's maybe cheaper versions to do that. Like for example, SNPs, like means single nucleotide polymorphism. Like everybody has, like I said, like 99% of the gene is same, right? So then no point like sequencing everything again and again, right? So we can only look at those like 1% that's actually we know are different. Then look at only the differences, right? So that will be the cheap version. So that's like if you want to look at the genetic variation. Yes. Yeah. 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 So yeah, question. Um, so right now AI is replacing a certain job. Like it cannot replace entire job. So it can replace certain task. Like for example, this AI code generation, like similarly GPT, like there's we code get integrated ChatGPT to the code writing process. So you can directly call it and just the code fragment generated, put it in the analysis. Of course, you have to verify what it gives. So they are like code generation, AI code generator that can replace certain tasks for bioinformatics. Like we don't have to like, I mean, we do the same thing. Like we look up in Google and Google it and then get the answer and try to plug it in and change something. That's how we code, right? Normally everybody do. So with the AI code generation, you can like make it easier to like automate certain part of the analysis pipeline, but it cannot do everything yet. So yeah, so I think it's an important development in like data science, like any data science, not just bioinformatics, like any data scientist toolbox, AI is playing a major part. Uh, Not, not really targeted because AI right now it's like general purpose. It's intended as general purpose. Like even bioinformatics things first started as general purpose. Like these tools didn't for bioinformatics. Like the were developed for bioinformatics. Like R Studio was not for bioinformatics. It's different. It's general purpose, but can be used in that. Um, yeah, I don't know if there's a specific AI tool just for the mathematics because it's like difficult, more complex than any regular day-to-day. So far, no. Yes. Right. Okay. So question is, when you identify a difference in one individual, uh, is like generalized, right? Is that what you want to know? So yeah, it's a statistical question, right? So you have two groups. One group has this mutation more than the other group, and we can find association for that disease because of this difference in the genome, right? So it's like a statistical problem. So not you can find one person has it, but maybe not just a random mutation. Might be nothing to do with, but it happens 10 times or 100 times. We have kind of association. So it's a statistical question. Yeah. remain reference. So like reference sequence is one reference sequence. Like that's the a human anywhere like use one reference sequence. Is that what you mean? Or yes. Ah, okay. So let's say if you're talking about the gene expression, genetic variation. Let's say like you have a different genetic profile than I am. And if you look at those like 1% of the genome that we know are different, we tend to vary between individuals, then you can look at this whole profile and see you, because let's say you have 10 million positions, can have variation, right? I have the same 10 million positions, but there's no way you get the same combination, right? Because I have there's different nucleotides to vary around 10 million positions. There's no way you can exactly replicate the same thing because it's unique for you, unique for me. So yeah, the position variation is different is enough to make difference between individual. Only like percent is enough. Yeah. Yes. Yeah. Yes. Good question. So yeah, validation, like it's a hard problem. Like you don't have all data you want in the world, right? So let's say in the example I showed, I was lucky because there was one group generated about 1,000 samples, same disease in a different lab, and another lab generated another like 600 samples for the same disease. So I was able to like do the training and thing in data set and completely leave the other set for the validation, like completely independent testing set. So that's actually the strength of that paper. It was completely independent. But if you don't have uh, the it's like you have 20 splits and you do it at times. Um, that that's the only way we can do cross validation. Yeah. Yeah, I can't hear. Can yes, yes. Okay, good question. So for the last like 10 years, the field of epigenetics hadn't considered the variability and the tissue specificity, like you measure circulation in blood may be different from any other tissue. So it was there, but there wasn't work research done on that. So with our work, we have discovered like certain regions in genome that are established like early in the human development, like first few weeks of the development, and then they are propagated through every part of the body. Like if you measure in brain or blood or any part of the tissue body, you have the same measurement pattern. So we have identified certain regions, about 20,000 regions. Those are like consistent. Like you measure it, 5 years or 10 years or 50 years, it's consistent. So that's the actual research that identifying those regions, they're finding associations for that difference in methylation to diseases. Uh, that was the problem before, so that's why there's research done on epigenetics to because epigenetics is hard problem because it's different from each other and it varies and there's an environment influence and all those things. Yeah, so that's why it's a very early stage of epigenetics. Uh-huh. Uh-huh. No, we are not working with cells. I mean, yes, it can I get like if they start with that, right? Yeah, there are people working in that, but our approach is different. So what we do is different approach. Let me see if I can have a slide. Don't have a slide. Yeah, so like here I'm showing like one of the region that we identified. So to do that, we took three tissues, like 10 tissues from 10 individuals from heart, brain, and thyroid. So if you look at heart, brain, and thyroid, they're like three lineages from the tissues equal, like mesoderm, ectoderm, and endoderm, right? So if you look at heart, brain, and thyroid, that from because all two type of tissues in the human body start from that. If you take three from that, 10 individuals, and we can see variation like here, this see, yellow color present one individual has same methylation values for this location. And this set of individuals have this variation values. This individuals have this. So we know this region see the variation between methylation, but that value is consistent within the individual. Same individual, the methylation is consistent. So this is basically like going back in time and see what happened during the early development period, and it propagates to now, because this data is from like post-mortem. After these people have died, they like decided to take the tissues and then they sequence this data. This individual like 60 or 70 years old, but the methylation is established at early development period, like when like first the development. And so our plan is to use these methylation patterns and associate with this diseases when you're 70 years old. Using the methylation patterns at early embryo. That's the question. Mm. Well, yes. So if we, I mean, I don't want to go into details of this. If you are interested, we can. I mean, we just take technical details. So if you want to go, we can go into this. Otherwise, we can like do more Q&A. So stop the sharing. I can share these slides. These are all like public knowledge, so I can free to share all these. Yeah. Yes. So for USA, there's something called DB database for genotype and phenotype. That's like everybody generated data, they put in one repository so the researchers can share. So it's only like to access to certain institutes because they are like data. So you have to have background like for like it should have access to getting that data. So there's a central repository in the US to do that. I don't think so. I mean, you can submit data from here. You can submit data. No restriction. I mean, they even welcome that because there's not enough data from South Asian or Asian countries in that. Most of the data is American or European. Yes, yeah, they can deposit if they want. That's allowed. So that's a good way to like even like preserve data. Like they will keep it forever, no charge, free of charge. Will keep the raw data. Yeah. Questions? I mean, there's no any questions. I can conclude the presentation. Yes. Yeah. Thank you for giving me the opportunity to contribute to science in any way I can. So, yeah. So there are so many other experts from Sri Lanka who actually want to do something but don't get a chance. So I think I'm lucky to at least do something. Yeah, and do something for the science community in Sri Lanka. Yeah. Thank you so much. Leave it? Okay.
Yeah. So with that, we can wrap up this session. And thank you so much, Dr. Chathura Gunasekara, for accepting our invitation and conducting this session. And I'd like to thank our audience. Earlier, it was mainly on site. We invited everyone to come here, but there were some requests from faraway places. So we created this link, and there's a Zoom community also joined. Thank you, everyone. And I would like to mention to all our audiences to reach out to NSF if I think you might have some needs to link up with expatriate Sri Lankan scientists for scholarships, for collaborative research. So we are most supportive, and we mostly welcome you to reach to NSF, and we will send our database right to those scientists and connect you with them. So please write to us if you have any such needs. Thank you so much. Have a good day.